All right, welcome. I'm running a little bit late today, so I don't want to, keep my student waiting. You know, I always try to at least attempt to stick to the schedule that I set for myself and agreed upon. But yeah, welcome, everybody and anybody. This is lecture number 3 of teaching biochemistry. And yeah, still getting back into the routine of streaming. I have, again, a lot of content planned. It's just a matter of like setting aside the time to actually do it. But still sticking to this content for the time being. And also, as I think I might have shared at some point, unfortunately and sadly, my grandmother, fell and she's in the hospital, but she's recovering, on the road to recovery. She had an operation and everything seems to be going in the right direction. But I've been focused a lot on that in terms of helping to take care of her and literally get her back on her feet. But once things have stabilized, I should be streaming much more because there's a lot of backlog of content. But again, I don't want to keep. I want to stick to the schedule, so I'm gonna get right into it. Just give me a moment.
Hello,
Hello.
hello. We do hate Goat Man. Fucking, except around here, we love Goat Man. Hail Satan.
How's it going?
Hello, good. How are you?
I'm all right. Running a little bit late. I apologize for that.
Oh no, all good. We were watching Leftist Brain Rot.
Oh, always good content.
Absolutely, everyone loves some fucking Leftist Brain Rot. Oh, actually, I'm not. I need to pop this shit out here.
Yeah, I need to do the same.
What the fuck am I doing? Yo, do you ever like do stuff and then you're just like, dog, how do I make it through everyday life? How do I even exist? And I just did it again. What the fuck am I doing? Oh my god,
I absolutely
dude.
have those moments. 1000%.
It's my fucking life. Ooh, my camera, my backup camera is kind of fucked, and by kind of fucked I mean it's not working.
Okay.
So I'm gonna have to virtual cam on to here.
Whatever works, whatever you feel.
Yeah, it's gonna look like this, unfortunately.
That's fine.
Okay,
That's fine by me.
Cool. And then we'll have the other screen and we'll make it work. But sick. Cool. Well then I guess you can see what's happening over here too.
I'm just gonna adjust some things on my end.
Hell yeah. We're gonna fucking adjust our dab counter on this end here while you do that. So bear with us here for uno momento. I'm gonna mute.
All right, you good to go? Oh, gonna have to wait a bit. No problem.
Oh, sorry, I was muted. Sorry, I was talking, but I realized I was in here. I'm ready. Sorry, we were watching a Ric Flair Woo compilation, but I'm ready.
Oh, never a bad time for that.
Absolutely.
All right, I think I'm. It probably helps if I share my screen. Actually, sorry about that.
Yes. No, all good.
Probably helps you a lot.
Tis quite crucial. Go ahead. There we go. Look at that, and now everything automatically pops back to the way it should be. Amazing. And we do still need to fucking figure out Video Ninja. But I've been so fucking busy, but it's been working great. And I will unmute the stream. Okay.
Okay, just making some last second adjustments, trying to get things, you know, as close to, well, not perfection, but oops.
Yeah.
All right. So, well, not picking up exactly where we left off last time. Okay, few slides back, just to like review and, you know, reinforce where we pretty much got through most of the, you know, whatever nitty-gritty hardcore chemistry stuff. Although again, I'm going to be introducing more chemistry as we go on an as-needed basis, and we're mostly covering now, you know, some of the fundamentals of biology that, you know, I think you need to know to hopefully start diving into biochemistry. But just for introducing, you know, what we're covering and the sort of order that we're going to go in is so this oh, actually I need to go back. Yeah, sorry, I want to start here actually, and then we're going to get into the textbook that we're going to follow next slide.
Yes.
Okay, but yeah, reviewing here the central dogma of biology. Okay, now just to reinforce and reiterate, okay, the concept is, right, we started with DNA. Okay, and the DNA is turned into RNA, ribonucleic acid, and that RNA is turned into protein. And these steps, okay, where the arrows are, they have a particular name in terms of the conversion of DNA.
Can I guess it?
You can guess it, absolutely.
Okay, it is I know they're translation and transcription, but like, translation would be DNA to RNA, transcription would be RNA to protein, or do I have it backwards?
Well, you got it backwards, but that's the common mistake, where DNA to RNA is transcription, and like, yeah, even totally, many people, myself included, sometimes like we have to second guess ourselves, like, what is the transcription?
That makes sense. Okay, sorry.
Oh no, it's okay. Keep going if you had a question.
And then do you want to pop up the next one, and then I'll kind of say why that makes sense to me. Because so like from like kind of like the stuff that I like the Bible-y things, like to transcribe the Bible, it would be to kind of just like, you know, to write it out and to like try to like just like rewrite it, versus to translate it would be taking it from like Hebrew to English, and like that's like the difference between like rewriting and like changing like it in a way that it's like different but it keeps the essence. I don't know, that's kind of how like my mind's thinking of it, if that makes any sense.
No, I think your logic is sound, where like so, you know, as well as we're going to see, so also probably today we'll get into a deep dive of like DNA, the structure of DNA, not just these cartoony graphics, but getting, you know, a much more detailed look at a molecular level of like what DNA is. But you know, DNA and RNA, as you're hopefully seeing from the cartoony graphics, that, you know, there's some similarities, as in one is deoxyribonucleic acid and one is ribonucleic acid. So, you know, it's not like a huge jump to go from one to the other. Like the essence, there's some essence still conserved, and so it's DNA to RNA, where it's still a ribonucleic acid, or it's still a nucleic acid, but one's deoxy and one's ribo. As opposed to translation, okay, a protein is totally different, like macromolecule, as we will see, we're probably not going to get into the deep dive into the proteins today, but probably next time, or at least building up, because proteins, as we're going to see, highly complicated, and well, pretty much a large portion of biochemistry is devoted to just like understanding proteins. But like, yeah, it really is. I think your logic is sound in terms of like, it's a translation of, we're going from basically a nucleic acid in RNA to something totally different, as in like, so, and that's where, okay, these arrows are pretty critical, where I think I mentioned last time that generally speaking, up until somewhat recently, okay, we always thought it was like one directional, as in you can only go from DNA to RNA and RNA to protein. There's like no going backwards. Well, we have since learned, and we've since sort of discovered, there's always exceptions in nature where, so you can go from RNA to DNA, okay, and that makes sense given the logic of like, some essence is conserved, so like just going backwards, not the huge deal. But as of right now, we have yet to find anything, and it would be actually a huge, discovery and benefit to humanity if we can go in the opposite direction of protein to RNA. We cannot do that directly yet. It's pretty much not done. And so the reverse translation can't do it yet. And so it makes sense that, we cannot reverse translate yet, but we can reverse transcribe, given what you've outlined. But yeah, go ahead.
So this is a question that chat asks, and maybe we'll get into more detail, but it's a question that like I kind of have too. But like, is protein just like a ball of RNA?
No,
or is it like
no, that's a good question.
Like changes and shit?
Yeah, so that's where it's a totally different, like almost like totally foreign to essentially unrelated, okay, to RNA. That's why it needs to be translated of like totally new language, totally different. The building blocks of, as you're sort of seeing here in RNA and DNA, the building blocks are nucleic acids, okay, and one's deoxy. We're going to sort of see like, what do we mean when we say deoxyribonucleic acid? We should be seeing that today. But yeah, since protein is totally different, like it really is like a new language. We have to go from a certain type of macromolecule, when I say macromolecule, like type of molecule of nucleic acid, to protein, where, as we're going to see probably next time, the building blocks of protein, okay, like the Lego blocks, are totally different. The amino acids, as we will see, as a little bit of a preview. But it's good, but to sort of also, maybe as a preview down the line, where I think their question was, well, is protein just sort of a ball of RNA? Yeah. Well, as we're going to see, like these things, as in you can get complexes, as in these things interacting together, as in you can have a ball of RNA sort of interwoven or intertwined with protein. Shit. We're going to see that there's a lot of mixing and matching that can occur. So it's a really good and pointed question, actually. But
this can get fucked.
Strictly speaking, proteins, like, it's a totally separate category to the nucleic acids. So moving from there, again, as I was about to say here, but yeah, we're going to be following mostly a particular textbook. It is this textbook, okay, but showing there, it's the 9th edition, but we're going to be following the 8th edition. And, okay, as maybe somewhat of proof of, I follow this textbook, is I have a version of it right here that hopefully you can see.
Beautiful. Oh, the black cover is sick with the silver.
They always, with different editions, they change the color, and I got the gray one. This is the 7th edition.
Oh, that's so
that
cool.
I have. I'm glad. Because, yeah, they've gone through red. Well, they pretty much, they rotate the colors.
Well, they're on the 9th edition. That's a lot of colors.
Yeah, exactly. But yeah, I agree. This brown. I'm glad that we're not going with the 9th edition.
Yeah.
All right, but again, like there was a runner-up. Choosing textbooks is actually contentious among biochemists. Yeah. A lot of people put this one as whatever, the Bible of biochemistry. But I don't particularly like. It's good. Like any standard biochemistry textbook is going to be good. But I'm biased towards this one because that's the one essentially that I was taught on.
I am a fan of Michael Koch's work. So.
Exactly. So again, a little bit of review. We saw this last time. So the beginning of biochemistry, understandably begins at the beginning of life. Okay, and what does the beginning of life look like. I believe last time you mentioned the Urey Miller experiment, okay? And I'm gonna prepare some slides on that because that's actually, I think, a good thing to go over. But I'm gonna reserve those slides that I prepare for a little bit later because, well, not that much later, like probably 2 or 3 weeks from now. Because those experiments are most related to amino acids, and we're going to get to probably amino acids next time. But
Sick.
all that to say, when did life begin? Okay, we have quite a bit of evidence, pointed to when the origin of life on Earth. Okay, we're right about here. Okay, about 3.5 billion years ago. We call it, well, microbiologists or other biologists, refer to it as Luca, the last universal common ancestor. Pretty much what we think, the first cell came into existence, or, maybe according to some people, popped into existence. But when it first evolved.
And when we say, like, well, the term "it," I think is really from an fucking fundamentalist creationist Christian in like mind. When we say "it," we like they don't mean like one cell, right? Or do they? Do they mean like one or like a population of these?
That's a fantastic question. More than you know. So that's why we phrase it like this, of last universal common ancestor, because we really, as far as we can tell, okay, yeah. I don't think we can ever really conclusively know, but we operate under the assumption that since it's the last universal, as in all life on Earth descends from one singular cell.
An individual, like it is a literal individual cell.
Yeah.
And then from that cell came all the other cells.
Yeah, and actually we're going to see sort of, hopefully it'll clarify in the next slide, because you're going to see sort of how we structure the tree of life, and like what's at the root of the tree of life is pretty much all signs point to the last universal common ancestor, or Luca, of like one single cell, and then evolution, takes off and we're gonna get branches. But then go ahead.
Also in that, and like the reason I bring it up, so Luca is literally an individual cell. But then when we say like something like our most recent common ancestor with apes is this, that is a population and not an individual. Right?
That's a good point of clarification. Exactly.
Okay. That's the distinction, because that is huge. Like that is huge. Like with the creationist stuff, like that. Well, if we all came from monkeys, why are there still monkeys? Type of stuff, which is a genuine question that many people still genuinely believe. And they think is a genuine gotcha.
Unfortunately, we're not going to get into much, speciation. Maybe at some point I'll try to work it in. But we're still going to, at a molecular level, look at evolution. And I think at the molecular level you get I think it's a stronger case for evolution, because we're going to see that, we're going to be looking at, DNA, okay, pretty closely, and we're going to see what DNA looks like in humans. We're going to see what DNA looks like, in, bacteria and E. coli, and, turns out looks pretty much the same. And there's many other similarities that we're going to see. But yeah, when we say last universal common ancestor, we really mean, at least all evidence point to one singular cell. And I think in the next slide, that sort of hopefully makes that a little bit more clear. So this is, a version of the tree of life. Okay, I'm not saying this is the tree of life. Well, maybe I should, reword my title. But we're going to see in the next slide sort of a different version. But we think, that last universal common ancestor, that one cell, it's pretty much right there at the root. And then from there, all other types of life, sprung or evolved into existence. We don't really, at least for our purposes now, okay, I don't really need you to pay attention to like Salmonella and Escherichia and all that stuff. What I mostly want you to at least, pay attention to is like, well, we're humans, we're right here, okay? Yeah. And maybe pay attention to the colors of like there's blue, there's black, and whatever, magenta, pinkish. And so in terms of the branches, okay, we're going to see that when it comes to like, categorization, like where do humans fall? Okay, humans fall in, the domain of what we call eukarya. And, if it's not clear now what it is, we're gonna hopefully be clarifying a little bit more in the next few slides. But all that to say is, here's where we are, okay? The last universal common ancestor is at the root of the tree. And yeah, humans are eukaryotic, and our cells are eukaryotic cells. And if you're asking what are eukaryotic cells, again, we're going to get to that, I think, in like 2 or 3 slides. But that's pretty much what I want you to take from the main message of this, where this is one version of the tree of life. We think that one for cells at the root, and here's where humans are in how we sort of like separate off, evolutionarily speaking, the branches. And yeah, bacteria and archaea are prokaryotic cells. We're going to be clarifying a little bit on what do we mean by what are prokaryotic cells, also in the next few slides. And how do we come up with the branches? Okay, well, that has changed over time, as in we weren't always aware of DNA. We weren't always aware of genetics, but pretty much as of now, like, we're mapping this out through, sophisticated genetic analysis, making the branches and the branching off. But it hasn't always been the case, as in, like, obviously 100 years ago, we didn't really know that much about genetics or DNA and chromosomes. And so that version of the tree of life looked different. But now I think we're getting closer and closer to, let's say, probably a more accurate tree of life. But again, this is one version, and in the next slide, I'll show you maybe a different version that maybe, provides a little bit of a different perspective, or just this tree of life in a different way. Where another version is this one, okay?
Ooh, that looks cooler.
That, yeah, definitely looks cooler, and maybe to some people, understandably, more confusing.
Yeah, absolutely.
But, okay, we still have prokaryotes, as in bacteria and archaea. They are prokaryotes. You have, okay, the last universal common ancestor, so that's what we mean of, like, that one cell, from that one single cell, everything, sprung from there. Or pretty much, again, that's the root. And so, yeah, go ahead.
Oh, sorry. Like, you'll probably answer this. Does that mean that Luca's a bacteria because it's in the blue and, like, life started out as bacteria?
That is pretty much what it alludes to. Of, like, we think we think Luca essentially looked like a prokaryotic cell, because as we're going to see in the next few slides, prokaryotic cells, we tend to think of them as simple, where from a certain perspective, if you're comparing it to like eukaryotic cells, so we, again, as humans, we're down here, we're eukaryotic, okay, it tends to be framed as, we are more complex. And there's also way more evidence to show, not just that prokaryotic cell was probably that last universal common ancestor, okay? And then this is essentially, trying to provide the information in terms of evolutionary relationships, okay? Where, yeah, the origin, we think, Luca, a prokaryotic cell, and that's why they're in blue there, the region there. And then hopefully you see that, eukaryotes, humans, okay, we're here in the orange-pink. And so, okay, based off, like, the length of the lines, okay, where, we had our first cell, a prokaryotic cell, and then through evolution, we got archaea, okay, and then somewhere down the line, well, you had humans, okay, or eukaryotic cells evolving into existence. And so, okay, this is trying to provide the information that essentially eukaryotic cells are closer, evolutionarily speaking, to archaea. And then, moving backwards, okay, we are further away, evolutionary we're still related, but we are more distantly related to prokaryotic cells. And if you see here, where archaea are kind of like, in the middle of the eukaryotic cells and the prokaryotic cells, archaea are kind of, at least they tend to be also framed as like a little bit strange, where they seem to be sort of like a hybrid of, like, they have some prokaryotic features and they have some eukaryotic features. And the way we've mapped it out, it's like, well, yeah, this evolutionary speaking through genetic analysis, they seem like they're like in between, like on the way to eukaryotic cells, like archaea, also evolved as kind of an offshoot, or on one of the branches of the tree of life. And we, even most, the microbiology that I've taken, we don't really cover archaea, just because, like, they appear to be pretty strange. Well, they do a lot of cool things, make no mistakes. Like, this is where If I remember correctly. I could be mistaken about this because my microbiology is a little bit rusty. But I believe a lot of archaea, you tend to find them in like very extreme environments, so
Bottom of
like
ocean shit.
exactly like really extreme heats, or really extreme like cold, things like that.
Oh god
Uh,
damn. So would that mean potentially, and the answer is probably I don't know, maybe, but that like places like Mars or like other planets where it's more hardcore temperatures, like if they had life, they might be more archaea
so
because it's extreme.
Uh, so I would intuitively, okay, the answer is essentially would like be yes, based off of what we observe, sort of here on Earth. And I believe at one point, okay, again, my microbiology is a little bit rusty, but I believe, okay, we gave them the label archaea, okay, because like, again, if I remember correctly, like they were viewed as sort of like archaic, of like, oh, these are like super like sort of like ancient, and they look like really strange. But then, once we had, genetic analysis, it's like, oh, well, actually, it looks like prokaryotic cells, go back further, and like archaea again. They're as I show here, they're in the prokaryotic category. But then they appear to have some sort of like eukaryotic features as well, where again, they seem to be sort of a hybrid. But like, I would say, like intuitively, you're right in terms of if we were to discover life on Mars, it would probably look either more prokaryotic like or more archaea like, and almost certainly not eukaryotic like. So just want to sort of put it on the table of the different trees of life. And as sort of a side note, okay, we're going to be doing a deep dive on, metabolism of like how do humans or eukaryotic cells like break down, sugars, carbohydrates, fats, etc., etc. And we're going to see that, many of the metabolic processes that we have as humans, like we literally find like the exact same or like 99% the same metabolic process in like, bacteria. And it just furthers the case of like the evolutionary relationship between eukaryotic cells like humans and bacteria. So what is a eukaryotic cell? What does that mean? Okay, when I say humans are eukaryotic organisms and we have eukaryotic cells, well, we're going to get into a brief primer of a eukaryotic cell, which is this. Okay, now obviously I don't expect you to like know everything here and like memorize it, but there's a few, let's say, key features that I want you to at least keep in mind. I'm trying to introduce them. I don't expect you to know them or necessarily recall them, and even then, like, I'm going to be reminding you of these things, and I'll probably use this same diagram just to like again reinforce the point. But this is a eukaryotic cell, okay, and it's understandably, if you're not used to seeing one, looks highly complex, which it is, okay, but this is not a cell biology course. But we're gonna be doing a little bit of cell biology, but this is an animal cell, okay, so when we're talking about eukaryotic cells, okay, even though those have sort of like a branch or like subdivisions or subcategories, where we have animal cells, okay, this little cartoon figure, somewhat resembles, okay, this textbook figure, as in this, the cartoon figure on the right, I just, in Canva, I searched, animal cell or eukaryotic cell, and this came up. And then, no surprise, like, the figure on the left, that's, like, straight from an actual biology textbook, and so they look pretty similar. But yeah, this is an animal cell.
It doesn't have the cum tail on it.
Yeah, exactly, which is pretty critical for some eukaryotic cells. But so our cells, since humans are animals, okay, our cells are animal cells, so ultimately we're eukaryotic cells, but subdivision animal cells, because as we will see, okay, at some point, another category of eukaryotic cell is a plant cell, okay, and humans are not plants, but basically plant cells, okay, they're under the overall umbrella of eukaryotic, but then different branch of plant cell, because as we're going to see, they carry out photosynthesis and they have specific things in their cells devoted to photosynthesis that we're going to see at some point. But I just want to introduce the concept of, a eukaryotic cell and an animal cell. And highlight some of the features, okay, where you don't need to read all the text here. One, because it's super small, and also second, because not critically important to understand, some of the fundamentals of biochemistry. Where at least I just want to highlight, okay, some things where, okay, you have a nucleus. I think most people are aware of the nucleus in a cell, or have at least heard of the term nucleus. And I think all I'd want you to know is that it contains your genetic information. That's where you find, your DNA, the blueprint, okay, the instructions that your cells need to make all the stuff that you need. And then you have your rough endoplasmic reticulum. Well, what is that? Abbreviated as ER, or rough ER. It's basically one of the major sites of protein production. Now we're going to see, okay, well, I introduced the concept of the central dogma of DNA to RNA to protein. And so since this is a major site of protein production, well, this is a major site of translation, of converting that RNA into protein, okay? They basically have a lot of not a lot, they have the machinery, okay, devoted to that translation process, which is a complicated machinery. But all I want you to really, at least keep in mind, but I'll be reminding, everyone, that the rough ER is a major site of protein production. And then you have the Golgi apparatus, or sometimes in some textbooks, they refer to it as the Golgi complex, okay? Fancy name, but ultimately, okay, in, the simplest terms, it's basically the shipping and handling of the cell. Of okay, even though cells are microscopic, they're small, just as things need to get around to your body, okay, in terms of circulation, things need to go to your liver, things need to go to your kidneys. At a cellular level, the cell needs to ship and handle things around. And the Golgi apparatus is pretty much, your shipping and handling department of, you go there, sort of, the shipping and handling facility. And then you have your lysosome, which is just as you have a stomach to digest things and break things down at a cellular level, okay, they also have sort of a stomach, or a thing that breaks things down. In addition to digesting, okay, things, it's also part of the stomach of the cell is also digesting, let's say foreign invaders, in terms of, as we're going to see probably at some point, that you have an immune system, and so you have immune cells, okay, and those immune cells, they try and go and digest foreign organisms that shouldn't be there. And so, some cells need a stomach, or many cells, such as a eukaryotic cell, needs a stomach, and the lysosome is basically the stomach of the cell. As in, let's say if I asked you, okay, I don't necessarily expect you to know, but if you happen to know, in your stomach, okay, do you happen to know sort of what's the major contents of your stomach that allows you to, break things down in your stomach?
There's acids and shit in there and shit. They're just stomach acids.
Yep, exactly. And inside the lysosome, it's basically an acidic environment. It's not, okay, it's not stomach acid, strictly speaking, as in stomach acid is hydrochloric acid, but inside the lysosome, there's totally, it's an acidic environment, okay, because it, like I said, it's essentially the stomach of the cell. And then, I think most people know, if I mention mitochondria, okay, it's the powerhouse of the cell.
Oh, yeah.
That's your major site, the critical site of energy production, okay? And do you happen to know, it's okay if you don't, I don't expect really anybody to know, but when we talk about, powerhouse of the cell, do you happen to know what mitochondria are making, the type of molecule that they're making?
The kind of molecule that they're making?
Yeah, as in it's the powerhouse of the cell. So what do you happen to know? I don't expect you to, but if you do, great. It's generating power, but power in the form of what?
I'm sure it's carbon based. I don't know.
Yep, you're absolutely right.
Carbon dioxide. I don't know.
You're absolutely right. Your instincts are right. That it's carbon based. It's called ATP. Now we're going to be seeing that.
Oh yeah, fucking chat said ATP. Yeah, you know me. You
Exactly.
down with ATP?
Exactly. We're going to be seeing that over
I'm
and
down with
over.
ATP.
Okay.
Okay.
Now we're going to see exactly what ATP is. It stands for adenosine triphosphate. I don't expect you to know what exactly that means or to visualize anything, but just introducing it now. When we say the powerhouse of the cell, we basically mean that it's producing ATP. And that is pretty much how we power our bodies, our cells, through the production of ATP. So that's a eukaryotic cell in a nutshell. And again, I don't expect you to remember this or like expect you to recall at any point. I'm going to be reminding you of these things when it's relevant, but I just want to introduce it now. And then I mentioned a prokaryotic cell. So what is a prokaryotic cell? So that's where we go to this slide.
Before we go,
Mhm.
what do the little fucking, I'm sorry for like not knowing, like the little cummy things, the flagella. That is
Yeah, yeah,
flagella.
exactly. You got it. Flagella.
Flagella.
Yep. Where that's basically, well, if you ever seen, let's say, a sperm cell, okay? So a prokaryotic cell is not a sperm cell, okay? Because
Yeah.
that's think of when you think of
Swimming?
prokaryotic cell, just think of bacteria and maybe if you can remember archaea, great, but pretty much just try to think of bacteria. But okay, it's pretty much a way to get around. It's kind of like pretty much it's a thing that whips around that generates
Little tail.
locomotion. Yeah, exactly. A tail, pretty much that can act as a rotor. Like literally, it can.
Helicopters?
Yeah,
It spins the phalanges. The flagellas.
we're going to see at some point that like there's an actual motor that spins there at a molecular level that acts as a rotor, okay, like a helicopter, and just allows it to propel forward.
Yeah.
Did that answer your question?
Yeah, that's cool. I'm excited to see the motor.
Yeah, because it
Proverbial
really
motor.
really is like a motor. It's actually pretty impressive.
Yeah,
But
that's
yeah,
cool. Okay, that does make sense.
but okay, pretty much the fundamental differences between a eukaryotic cell and a prokaryotic cell are essentially, highlighted in like the first point of like they don't have a true nucleus, as in going back to this one, okay, this big purplish thing in the center, this giant thing, that's the nucleus. That's what we have in our cells. We have a nucleus that contains our genetic information. Okay, the prokaryotic cells, this is why I say they're very often framed as like they're more simple. Like they don't have these, nice compartments. We refer to them as membrane enclosed organelles, okay? Where just as humans have organs at a cellular level, okay, your eukaryotic cells have organelles. Your mitochondria is an organelle, okay? That Golgi apparatus is an organelle. Analogous to, like, again, kidneys, stomach, at a cellular level. But prokaryotic cells, they don't have, these membrane enclosed organelles. That's fancy terminology of like they're not highly compartmentalized, or they don't look, they're not obviously highly compartmentalized. They don't have that true nucleus. They just have like this like blurry region in the center. There's like
They're just
there's
floating and chilling.
Exactly. Where if you look
They're
inside,
a place to be.
If you look inside, look at the cross sections, like there's just like a bunch of stuff in there.
Yeah.
It's kind of like floating around. Okay.
Yeah.
And that's why I sort of mentioned here, like they're often thought of as, and taught, also it's taught to me as like simple and less organized, which
They're just chill.
At
they're
some
the stoner
level,
of the cell.
Yeah.
They don't really have much going on, but they're just hanging out. They're having fun though. Like we can't judge them.
Yeah, exactly. We shouldn't judge them. And in actuality, okay, prokaryotic cells are actually, they're complex. Okay, E. coli, that's a bacteria. Okay, so they're prokaryotic cells. They've done a lot of work on E. coli, and they do actually have organization. Is it as organized as a eukaryotic cell? Well, no, I guess technically not, depending on how we want to define, organization. But yeah, they don't have these nice compartments of like, here's your mitochondria, here's your nucleus. But they are organized. They're not just this floating sack of stuff that has a flagella that spins around.
There's more to them.
Exactly. They are more than meets the eye. That's what I
relate to the prokaryote tick.
Exactly. I think we can all root for, the underdog to some extent.
Absolutely, that's my boy right there.
And again, just to bring in some of the research, okay, and to sort of prove my point that prokaryotic cells, there's organization. They're often overlooked. They're not again this floating sack of stuff. Well, yeah, we don't need to dissect this paper. I just want to highlight, okay, that they're talking about bacterial cell replication, and I just highlight that spatial organization is a hallmark of all living systems. And even bacteria, okay, bacteria are prokaryotic cells. The smallest forms of cellular life display defined shapes and complex internal organization, showcasing a highly structured genome, cytoskeletal filaments. You don't really need to be concerned with that right now, but we'll get to that. And they have, scaffolding structures, a bunch of scientific jargon. But all that to say is, like, there is some organization to them. Maybe not as organized as eukaryotic cell, but they're complex, they're organized, don't overlook them. That's
Cytoskeletal filament. That is a fucking, that shit's gnarly.
Yeah,
that's a gnarly term.
And we're going to get, at some point we're going to do a deep dive on what that is. That's also just fancy terminology. Just as you as a human, okay, you have structural rigidity. Okay, as in you can stand up upright, and what provides structural rigidity and integrity to you is your bones, your skeleton. And at a cellular level, okay, even though they're microscopic and they need some sort of structural rigidity. And that's why we call them cytoskeletal filaments, derived sort of from skeleton, as in they're providing structural support and integrity to the cell. They do other things as well, but one of the critical features is providing there is.
It's kind of like on like a 3D printer where like you're fucking making the thing and it's got the little scaffolding fucking structures that you like eventually break off, but to keep it nice and straight and good.
Exactly. So we just get rid of. Okay, sorry about that. So okay, we've gotten a brief tour of a eukaryotic cell and a prokaryotic cell, and as usual, you're ahead of the class, okay, because when I started off on that first slide, okay, I'm just gonna go to the question. And, let's see if you can repeat or you kept in mind what we just went through. So a little bit of a quiz, multiple choice. Let's see, one moment.
All right.
Cool.
I'm going to have a rough time with this one, but I think... I know the central dogma.
So is it DNA is transcribed into protein, and protein is then translated into RNA? Is it DNA is transcribed into RNA, and RNA is then translated into protein? Or is it DNA is translated into RNA, and RNA is then transcribed into protein? Or, lastly, is it a fantasy comedy movie directed by Kevin Smith starring Ben Affleck and Matt Damon?
Ooh, that's just dogma, I believe. I believe D is just dogma. But it is DNA is transcribed into RNA and then translated into protein. My eyes are closed. I didn't look at the thing. Whichever one that one is.
Yeah. Well, do you want...
it is... Okay, it is... DNA is transcribed... B. It's B. And the RNA is translated into protein.
You are absolutely correct. It is...
Yes.
DNA is transcribed. So, again, I'm pretty big on this. I'm going to be sort of reiterating it over and over. Because even at, advanced levels, some people understandably get it mixed up. Transcription and translation, they sound similar, okay? There's a lot of overlap, and it's describing, a process. But, like, it is pretty important to keep it in mind, and I would say it's important to, like, orient, I like to use it personally as, like, a compass of, like, well, we're talking about proteins, okay? So at some point, this protein was made by translation, okay? And we're going to see, like, what translation looks like. We're going to do a deep dive on proteins, okay? And then, okay, next question. Actually, got 2 in a row.
Let's go.
Should be probably, I assume, pretty easy for you.
This is easy. 6 to 10,000 years. Depending on how you do
1.5 billion years ago? Yeah. Is it 6,000 years ago?
B.
4.5 billion years ago? Or 3.5 billion years ago? You're going with B?
Well, B is what I was raised with, but I think it is D. Is that true?
Is that your final answer?
Fuck yeah, I believe so. And like the universe was like 4.5 billion. No, is it 1.5 billion? If that is that like the Earth? Like, am I confusing like the creation of the Earth with the fucking
Yeah,
yo
yeah. So we're talking about where did Luca begin?
yo Luca 1.5?
Final answer?
Yes. Oh fuck. Oh god damn. No, the Earth was 4.5. So is that true? The Earth was like 4.5 or like 4.8 or some shit?
Even
Or
I'm a
am
little bit
I-
rusty on that. It might be 5 or 6. Because well, in biology, we're not really concerned about where Earth came
damn.
into it. We're more like where did Luca come into play?
Yeah, we literally just went over this, but yeah,
No,
fucking goddamn the fucking Earth fucking thing, and I was like, wait, is that the universe? And I'm like, wait, no, wait.
I know.
Earth is 4.5 billion years old. Paul Squall has confirmed in chat.
Yeah,
Thank you.
intuitively,
The universe
sounds
is
right.
14 billion. That's it. 14
Well, yeah.
billion.
So yeah, 14
I grew up in
like
a cult, I'm
universe. Or then don't apologize. Yeah, if anything, you know, some of your family should be apologizing to you. Personally.
We've had that talk.
I'm hoping it was positive.
Yeah,
So,
we've all learned and grown.
So that's this is some fundamentals of biology in a nutshell. Okay, now we're going to do a deep dive, a fairly deep dive on DNA. Okay, you know, well, one of the important molecules relevant to life. You know, as we just saw in the eukaryotic cell, your DNA is in your nucleus. Okay, pretty much, you know, it's like right smack in the center of your cell. It's, you know, enclosed and all that stuff. So, but before we get into DNA, okay, we need to cover one thing, which is like, you know, we're going to be looking at molecules, okay? And they're going to be looking, you know, maybe to some people, understandably, like complicated, maybe a little bit overwhelming. And, you know,
You're glitching a bit and getting a little robotic. I think you're dropping frame. Your screen share is good, but your camera's glitching a bit here.
One second.
You look smooth on your side.
Testing. Better now? Worse?
Yeah, it sounds better, but your camera's frozen. And I can see, like, I'm watching your stream, like, moving my head. On my Discord, it looks smooth, but I see it on, like, your screen, it's looking like it's glitching. But your screen share is good, and your voice seems fine.
Okay. Yeah. I see it in my OBS that your camera.
Is it me?
No. Like, I think it's on. It seems. Well, yeah.
Let's see if I'm dropping frames here, or my bitrate's good. That should be good. Should I back out and come back in? Here, let me back out and come back in. It'll be 2 seconds.
Yeah. No problem. Take your time.
Oh, you're good now.
Oh. Okay.
Okay, let's just let it rock. You're perfect.
Yeah. And you're good now too.
Okay. Awesome.
All right. So. But yeah, we're going to be looking at molecules, okay? And for some people that aren't used to looking at molecules, it can be a little bit, even for myself, you know, when I first was looking at them, it was like overwhelming, okay? And it's hard to like piece things together. And, you know, it takes some getting used to, but I have no doubt that you'll get used to it. But
let's go.
1 second, let me go to the next slide. Okay. So, as mentioned, okay, scientists are lazy. Okay, so we take shortcuts when drawing chemical and molecular structures. Okay, so what I'm showing here is the same molecule depicted in 3 different ways. Okay.
Oh fuck.
On the right, okay, you know, that's, let's say, you know, you're correct showing every single atom. Okay, you have your carbons and hydrogens. Okay, but what it can get tedious, and so this molecule called pentane. Okay. It's just shown differently in all 3 figures. Now, if you want to venture a guess, I don't necessarily expect you to know, but why do you think we call it pentane? What does pentane?
Because there's 5 carbons.
Yeah, that's exactly right. Or at least the penta part is why we call it pentane. There's a reason why we call it, instead of, you know, not just penta, we call it pentane. But the penta part is derived like it's 5 carbons, as in, what do you think if you can, let's say, follow the convention by intuition? If I have 6...
octane!
Yes.
So it'd be 8.
Yeah, exactly. Octane
like
is
motherfuckers
8.
got that high octane ass shit. Okay.
Now if you can extrapolate, what do you think 6 carbons would be?
6 carbons. Hex?
Yeah, exactly. 6 carbons is hexane.
And then hept. Oct.
Yeah.
Niften. If
the 9 one is weird. We call it nonane. Nonane.
That's pretty sick.
But that's pretty much like the convention. Okay, this is getting into a little bit of the weeds of like organic chemistry, but there's obviously huge overlap between organic chemistry and biochemistry. But like, yeah, it's pretty much, since there's 5 carbons, okay, and we call them, okay, as in these carbons, okay, are saturated, as in, what do we mean by saturated? As in they all have the maximal amount of hydrogen, okay, bound to them. Where, I don't necessarily expect you to recall, okay, but carbon, okay, do you happen to recall how many bonds does carbon like to have?
Yeah, carbon likes to have either 3 or 6. I think it's 6. Or not 3. It's 3. 4.
Yeah, count on the right figure.
Count
how many
on the right figure?
Well,
4.
I mean on the right hand, exactly, as in carbon.
Yeah.
All of them, okay, have 4 bonds. As in, if you look at the carbon number 1, okay, I numbered them, where it has 4 bonds, okay, 3 hydrogens, okay, and then connected to 1 carbon, okay, so that's 4. And then obviously if you go to the middle carbons, okay, it's 2 hydrogens, okay. This is what I mean by saturated, in terms of you have the maximal amount of hydrogens there. Can't add any more. Carbon will not accept anymore. You're not going to find, at least under the most extreme circumstances, at least not in biochemistry, we're not going to be finding carbon with 5 hydrogens bound to it.
Oh, so that would be gnarly.
So since we know that carbon, okay, it likes to have 4 bonds, okay, pretty much we try to do a shorthand of what we see on the left. Of if I just have this one there, okay, if I draw it like this, of just 5 lines, I can sort of deduce, well, carbon number 1, I can just I can picture in there's 3 hydrogens there, as in we're going to see ways of what it looks like what if there's 2 hydrogens there and whatnot. But either way, if it's just a single line like that, pretty much like this, 1, 2, 3, 4, 5, it's every carbon there has the maximal amount of hydrogen. The one on the ends, the 1 and 5, they have 3 hydrogens, and the ones in the middle, they all have 2. Okay, and that's our shorthand, and sort of the one here, that's sort of maybe our compromise of a CH3 and then 2, 3, 4 and then CH3.
I was gonna ask why the difference, but that makes sense. Yeah, because the ones in the middle only have 2, because they gotta make double bang on either side. They're getting it from both ends, and then that they don't have as much. Okay.
Exactly. So in biochemistry, we're probably gonna more see sort of like this, as in when it's CH3, you'll probably see it. But every once in a while, you'll come across just a bunch of lines. Well, if it's a bunch of lines like this, and this is drawn a very specific way. It's not the lines are not just randomly oriented. They have specific structures to them. But either way, whenever you see something depicted like this, you can just fill in of there's a bunch of hydrogen there. Well,
so you just assume that each end of the line, so be at the bottom, the top, the middle, as it goes dingle dingle dingle, there's a carbon in that little blob. You just assume that and know. And also assume that hydrogen is there too. You're not thinking that it's anything else.
Yep, exactly. Good point of clarification. So whenever you pretty much see that, and pretty much this is a touch of organic chemistry 101, where whenever you just see these staggered lines, okay, it's a carbon, okay, and it's a carbon unless otherwise specified, and we're going to see that there will be times where it's otherwise specified. We're going to see sometimes nitrogen there, but the nitrogen will be depicted. But whenever it's just a bunch of lines, like that, just fill in carbon. Exactly. So, with that in mind, okay, starting with doing the deep dive on DNA, okay, and what I call a primer on DNA. And there's a reason why. We're going to discover why I'm calling it a primer on DNA. There's a little bit of a nerdy pun there. I don't expect anybody, except for, a hardcore biochemist or molecular biologist, to understand why I call it a primer on DNA. But maybe with, not maybe, in time you will maybe appreciate the nerdy pun. Or you
I hope to get the pun. That's all I want to get out of this, is be able to appreciate puns and to like rid myself of my creationism that I still fucking don't even know I understand.
I'm gonna do my absolute best. But yeah, the nucleotides are the building blocks, okay, of DNA, where pretty much, so I'm sure just about everyone, yourself included, has heard of the term polymers, right?
Mm-hmm.
Okay. Well, to make a polymer, well, poly means, you know, many, but to get up to many polymers, you need monomers. So all I'm saying, this is like fancy jargon for like, we assemble monomers into polymers, okay? And nucleotides are composed of 3 components. What are those 3 components? Those 3 components are a pentose sugar, okay? And if it's not clear what that is, hopefully it will be clear, but obviously you can ask questions at any time. And then another, okay, component of the nucleotides is a phosphate. And again, it's not clear yet. It should be clear pretty shortly. And then there's the nitrogenous base, okay? And the nitrogenous base, those are sort of like the critical, okay, components of DNA. Okay, now what does all this mean? Pentose sugar, phosphate, nitrogenous base, okay? Well, starting first, okay, we'll get to the pentose sugar and the phosphate, okay, pretty soon in the next couple slides, but I want to focus in on the nitrogenous base first, because they sort of like, you know, they ultimately what make DNA really matter, as in when we're talking about genetic information, we're really ultimately talking about the nitrogenous bases. So what are the nitrogenous bases? Well, they're shown here, okay? And that's why I want to start with, you know, some of the organic chemistry of how do you read all these lines, okay? Or anytime that there's a line, unless otherwise specified, okay? And they're specifying here, where there's an N, it's a nitrogen. Where there's an O, there's an oxygen. But anytime that you see like, you know, a point, okay, or just like a line ending, there's a carbon there, okay? But these are your, and there's a reason why we call them the nitrogenous bases, because there's some nitrogen there, okay? And so, you have your adenine, okay, you have your cytosine, your guanine, and your thymine, okay? These are your nitrogenous bases, and I'm essentially introducing them now, okay? But we're going to be seeing these at least over and over a number of times over the next few slides on this primer of DNA. But this is just one of the components of nucleotides. So we're trying to assemble a nucleotide, and the nitrogenous bases are one of the components, okay, of the nucleotide. One of the 3 components, okay? But building up, okay, a little bit more. And when we're talking about, okay, and we're going to see this a little bit more in the next few slides, when we're talking about the sequence of DNA, of getting your DNA sequenced, we're talking about the sequence of your adenine, your cytosine, your guanine, and your thymine.
Are those the little bars in the fucking spinny thing? And
Yeah,
they say like CGAT?
Yeah, we're going to get to that pretty much, I think, in about 1 or 2 slides. That's why, if you look at the words, you have adenine A, cytosine C.
Yeah.
That's the actual, your A's, your T's, your C's, and your G's. We're going to be seeing that over and over. But yeah, those are your nitrogenous bases, okay? And so, okay, how are they? We're starting off hopefully simple, and we're going to be building up from here. And so, here, okay, you have your pentose sugar, well, pointed there in, you know, pinkish red. You have your phosphate, okay, and then you have your nitrogenous base, but they're just calling it base 1, base 2, base 3 here. But pretty much you can fill in what we just saw in the last slide. Well, again, I'm going to show it in more detail as we go, but I'm trying to start off simple and build up to the complex. But all that to say is pretty much the main message is sugar and phosphate form the backbone of DNA. Now, what does that mean? 1 second, I'm answering a question from my own chat. Sorry about that. But I just want to introduce that there's a backbone to DNA. Just as, you know, hopefully humans have a backbone, and you have a backbone, I have a backbone. Pretty much, you know, what provides the structural integrity, okay, to the DNA. But again, I'm going to be providing more detail. I just want you to introduce this concept here. You have your 3 components, okay? You have your nitrogenous base, you have your pentose sugar and your phosphate. Okay, so what does it look like when we start trying to assemble them? Start assembling them together. So it looks like, okay, everyone knows as sort of the double helix. Okay.
Yeah.
So I like to think of it. It might be a little bit faulty, but if you take a ladder, okay, and twist it, okay, and that's sort of pretty much in my mind what a double helix looks like. As in, you have on the outside your ladder, okay, the backbone, okay, on the outside on the perimeter, and in between the rungs of the ladder are the nitrogenous bases. And again, we're going to see, we're not at the molecular level yet. I just want you to get a sense of, the overall structure and get a sense of, like, there's a sugar phosphate backbone and there's nitrogenous bases there in between. Okay?
Is one side, like the purple, is that sugar and the blue phosphate? Or is it like it's like a mix together?
Yeah, so it's an excellent question. So it really is, okay? It's a pattern.
Oh yeah, it's like that. That's yes. Okay.
Exactly. So it's phosphate, sugar, as in what we're seeing here, we're seeing just one, if you take the ladder and cut it in half, okay? We're seeing just one half of the ladder. This is not depicting the double helix. As in, to fully depict the double helix, you have to sort of like copy and paste. We're going to see it, but all we're seeing is like one strand.
Yeah.
So, but yeah, so that sugar phosphate backbone, so that like those two ribbons, you could think of it, not think of it, it's sugar phosphate as repeating units. So we're going to see that, and again, we're going to actually zoom in at a molecular level. I'm just trying to get, trying to start with the cartoony figures, just so you get a sense. Yeah. And then we're starting off, zoomed out, and we're going to zoom in even more. And also, importantly, this structure of DNA was discovered by Francis and Crick in Watson and Crick. Sorry, James Watson and Francis in 1953. So almost exactly 79 or 16. 70 years ago. My math is totally way off. But either way, a long time ago. And we're going to I'm going to show you in a few slides like what their original research looked like, and it's staggering. At least in my mind, it's staggering. Like how they were able to see like this double helix.
Were they like actually able to see it, or like in their head? You mean like imagine?
That's a good question. I'm going to show you what data that they were looking at, and then you're going to it is kind of mind blowing of like the data that they looked at, and then from there they were able to extrapolate, okay, like, oh, this double helix that like 70 years, like we know pretty much absolute certainty that's what it is. Okay.
God damn, dude.
Let's go. But yeah, James Watson and Francis Crick, very important. Although I think one of them has since sort of like gone off the deep end. I think it's
like crazy Chud deep end, or like fucking everything gold.
No, crazy Chud, like, sort of, I think they started spouting
gender critical nonsense.
And like, IQ, intelligence, race stuff. All that. Well, since they discovered DNA, they think that they're, the absolute expert on everything DNA. But either way, that's an aside. At the end of the day, okay, their contributions are important, despite the fact that, in my opinion, one of them has gone off the deep end.
Yeah.
So, getting a little bit, assembling a little bit together, okay, where now, we're flattening it out. We're not, so this is still a double helix. So this is why I say, to me, it looks like a ladder, as in, if you twist this figure on the left, you pretty much get what we saw in the last line, that double helix where, that sugar phosphate backbone, okay, your teal or green lines, that's your phosphate sugar backbone in repeating. And again, we're going to see that soon, pretty much in I think 3 or 4 slides. But yeah, as you mentioned earlier, that series of A's, T's, C's, and G's, okay, the rungs of your ladder, that's your adenine, your cytosine, your guanine, and thymine. As in, you have your C, cytosine, your G, guanine, and your adenine, A, your thymine, T. And as they're sort of alluding to here, okay, you have a C interacting with a G, you have an A interacting with a T, and I'm going to be reminding you of that pretty consistently. But just keep in mind that under healthy circumstances, C interacts with G, and A interacts with T. And we're going to see in a little bit, well, what do I mean by interact, okay? But they're sort of here fitting together like puzzle pieces. But they actually, they make actual connections with each other. But yeah, so again, we're not at the molecular level just yet. We're still mostly, the simplified figures. But just recognize that the sugar phosphate backbone on both sides, okay, those on the outsides of the ladder, it's just phosphate, sugar, repeating, okay? But you're going to see that pretty soon. And then, okay, when getting into what I mean by how do these A's and T's and C's and G's interact, this is what's occurring, okay?
Oh, shit.
And that's why I introduced, okay, covalent bonds and hydrogen bonds earlier, because here is where they are playing a part. As in, okay, you have your adenine, a nitrogenous base, okay, it binds with thymine, okay, and this is how they bind, and that those dashed lines, those are hydrogen bonds, okay?
And just to be clear here, the covalent bond is like the bond through like the covalent, or the valent electrons.
Yeah,
exactly. But the hydrogen bond is a type of dipole-dipole reaction.
Yeah, yes, perfect. Yes, nailed it.
Fucking, I remember the thing, and I'm so fucking proud of myself. And I'm gonna take another fucking dab in honor of that for any haters who say I'm too stoned to fucking understand things.
Absolutely, no.
I did a science.
That is perfect recall.
I remembered.
Covalent bond, that's that sharing of those valence electrons, and the hydrogen bond, okay, it's that special type of dipole, where and hopefully, okay, but we're going to be seeing hydrogen bonds over and over and over again. Yeah, but hopefully you're seeing a little bit of a pattern of you have a hydrogen, okay, in all circumstances, sandwiched between, okay, two electronegative atoms, either an oxygen or nitrogen, or both oxygen, both nitrogen. Where pretty much look at the adenine and the thymine, the dashed line, you have hydrogen, and on the right is an oxygen, on the left is a nitrogen. And then go down the second one, the other hydrogen, you have, well, two nitrogens on the side. And it's the same theme with the guanine and cytosine, okay? And then, okay, another thing to point out, in terms of hydrogen bonds, okay, do you notice any differences between A and T and C and G?
I noticed that there's 3 as opposed to 2.
Yeah, that's it. You answered exactly what I wanted. Yeah, that's it.
Oh, nice.
That is actually relevant. We're going to see that at some point.
Does that make it stronger because there's more bonds?
Yes, exactly. As in to pull C and G apart, it takes more energy.
You gotta fuck around harder to find out.
Exactly. And this has ramifications not only for research purposes like PCR, as in, well, everyone, I think at this point on the planet has heard of PCR when it comes to COVID and PCR testing. But taking into account what we call the GC content is actually important when it comes to designing an appropriate PCR experiment or protocol. If ever at some point you come across GC content terminology, they're talking about, well, how much C's and G's are there, because you have to take into account that there's actually 3 bonds there instead of 2, and so it takes more energy, more heat, generally speaking, to break those bonds or pull those things apart. But yeah, that's pretty much one of the key differences to keep in mind. Where, as you correctly pointed out, the bonding between C and G is stronger, okay, than the bonding that occurs in A and T. So, zooming in, okay, a little bit more of okay, again, we're not quite at the molecular level, but still, how is the double helix formed? Okay, so I'm talking about, complementarity. And I sort of introduced this earlier of A binds with T and C binds with G. And it forms those bonds that we just saw on the last slide, those hydrogen bonds between A and T and C and G. Don't let on the right hand side those dashed lines, okay, mislead you. It's not one-to-one. They're just simplifying it to just show that C and G are binding.
I'm just gonna ask.
Yeah, that's a good point of clarification. But this is how, let's say, the two strands come together. You have, one strand that's complementary, as in on one strand, okay, and follow the arrows. As we're going to see, there is actually directionality, okay, to each half of the DNA strand. Where if you follow the arrows, okay, reading from bottom to top, following the arrow, it's, C, G. Basically, anytime that you have a C, you have a G on the other strand. And anytime that you have an A, you have a T on the other strand. And, vice versa. But basically, A with T and C with G. And so they spontaneously, or they will, under, like, most conditions, like, they will spontaneously come together. As in, we can break these bonds. If you stick DNA in a tube, okay, in water, and you heat it up with enough heat, okay, you'll start separating the strands. As in, you'll go from the right hand side, double helix, to the strands separating. Okay, that's actually what happens in PCR. Of like, you raise the temperature, and we call it melting DNA, of breaking those bonds that we saw in the last slide. But if you decrease the temperature, okay, and you just bring it back to room temperature, it will spontaneously assemble back, okay, as into how it was, making those appropriate connections of the A's and T's and the C's and G's the vast majority of the time. It's actually pretty incredible in terms of, like, you can literally have like millions and billions of base pairs, okay, that's, in your double helix. You can separate them, okay, through heating them, and then if you just cool it down, all those billions of connections will just spontaneously reassemble pretty much perfectly in most scenarios. But yeah, all that to say is complementarity, and they spontaneously assemble to form that double helix. Now,
oh, sorry.
No, go for it.
Can the letters? And I know they're not letters. Like, I know that they're not, but like, can they just are they random? Or are they always in C G A T T A? Like, are they always in like a specific order? Like on the left hand side.
When we're talking about genome, okay, like the human genome. So let's say, it'll be unique to you, as in virtually, okay, not actually 100%, but like in virtually every part of your cell, okay, you have a genome, okay, so you have your own series of A's, T's, C's, and G's that's unique to you as a human. Obviously, it shares some commonalities with other humans, but unless you have an identical twin that you don't know about, or I don't know about, you have your own unique sequence of A's, T's, C's, and G's.
Oh shit.
And so it's random in the sense that, like, it was shaped through evolution, which is all, there's a strong element of randomness in evolution, so to speak, in terms of like how evolution like works at a molecular level. And we're going to get into mutations of like, what does it mean to, mutate your DNA? As a preview, when we're talking about DNA mutations, it's like, you're changing one of your A's, T's, C's, and G's. You're making an actual like mismatch, so to speak, of like, well, instead of an A, you have like a G there, or a C. But we'll see that in due time. But like, all that to say, it's like your DNA sequence, your series of A's, T's, C's, and G's is unique to you, unless you have an identical twin. But obviously, again, you share some similarities with other humans. But your own personal DNA sequence, your series of A's, T's, C's, and G's, will be unique to you. And so, at a molecular level, okay, what does the DNA look like? Okay, let's try to put it all together and look like at a molecular level what the DNA looks like. Okay, the double helix. And so, okay,
I'm nervous.
Looks like this.
Oh,
There's
that's
two
cool.
depictions here, okay? And I've tried to, you know, point out, okay, some of the things that I've introduced earlier. The sugar, the phosphate, and the nitrogenous bases, okay? So, you know, you have your double helix here on the left, okay? And so in blue, okay, your blue ribbons, that's your sugar phosphate backbone. And when I say sugar phosphate, it's like it repeats a pattern. You can see the pattern there on the right of like pentose sugar, phosphate, pentose sugar, phosphate, pentose sugar, phosphate, and it's pretty much the same on both sides. And so, you know, spiraling up on the double helix, okay, on your backbone, you have your sugar phosphate backbone, and then in between, okay, you have your nitrogenous bases, okay, binding. As in, you know, you can maybe fill it in here of like what the arrow's pointing at. It's a nitrogenous base. You have an A on one side and a T on one side, and it looks like this at a molecular level. Okay, that's why I'm saying, like, if you take a ladder and twist it, at least in my mind, okay, it looks like a double helix. Like, on the outside is like, you know, the bars of the ladder, and then in between are the rungs, and the rungs are like the nitrogenous bases, where hopefully you can see, or maybe like a spiral staircase, so to speak. But like a double spiral staircase. So that's what happens, and that's pretty much what DNA
Double
looks
spiral.
like at a molecular level. Where again, you have your A's, your A binding with T, with those two hydrogen bonds in between, and then you have your G binding with C, with those three hydrogen bonds. And then you'd have to extrapolate it. When the human genome, there's billions of them. It's 3 times 10 to the 8.
Jesus.
So it's a lot. But that's ultimately, okay, what DNA is at a molecular level, looking at it, you know, at the atomic level. And so, okay, maybe wrapping up in terms of, you know, how was this discovered? As mentioned,
Question before we move
Absolutely.
on here, just to this diagram. Are the 3 and the 5s at the top and bottoms of the thing relevant?
Yes, they are. So there's directionality. So those are referred to as 3' and 5', as in like 5 in that, you know, little whatever dash or a little check.
Apostrophe.
Yeah, exactly. So, you know, in molecular biology terms, so there is directionality to the strands where, so think of it this way. If you read both of them from the top, okay, following the convention of sugar phosphate, okay, what does the left one, what's the pattern on the left?
What's the pattern on the left?
As in, if you're just using the convention of sugar phosphate, as in if you start off like, what's this first thing here?
So it's phosphate, sugar, phosphate,
exactly. And then on the one on the right,
then it's sugar, phosphate, sugar, phosphate.
Exactly.
Okay.
But if you read 5' to 5', okay, what is the pattern?
Oh, then it's phosphate, sugar, phosphate, sugar.
Exactly. So that's us trying to give directionality to the strands and orient it, because that actually has relevance.
So that's essentially saying, okay, like in my little fucking brain here, you got fucking left side, he's coming up, and then Buddy's like, oh shit, let's go, and fucking flips, and they flinkle dick.
Yeah, exactly.
Oh, because like it has a... Okay, cool. One's going one way and one's going the other.
Exactly.
Is it always one way and the other, or not always? And that's another thing that we'll get to later.
No, it's pretty much, well, in healthy, typical circumstances, it is that direct of, one's 5' to 3, like, again, this is all arbitrary. It's sort of like saying one's north to south and the other one's south to north, or one's 5' to 3', and one's
up to down and one's down to up.
Exactly. Or, you know, one's west to east and one's east to west. But, like, that's us trying to, like, you know, orient ourselves where this does actually have ramifications, okay? We're going to, when we're talking about DNA replication, okay, when your cells want to divide, they have to duplicate their genome. Well, how do they do that? There is machinery, okay? When we're talking about, like, and your machinery, when it wants to replicate your DNA, it has to, like, scan your DNA. And as we will see at some point, like, it scans your DNA in a very specific, like, way, as in, it seems to do it in a specific direction. As in, it looks like the machinery, it's like it's going to take one strand and it's going to read it in like the 5' to 3' direction, and it's like never going to go in the 3' to 5' direction.
It would be like if a fucking barcode scanner and like suddenly instead of like vertical lines, it's sideways. Like the fuck are you doing, dog?
Yeah, exactly. Where it seems a little bit trivial, understandably. Even when I was first introduced to it, it's like, why of all things, 5' to 3', where it's like, yeah, one's phosphate sugar, and the other one's sugar phosphate. It's like, well, who cares? But like, no, it actually seems to have actual ramifications, that your cells themselves take into account. But yeah, great question. So yeah, those 5' and 3', they are relevant. But again, we sort of like arbitrarily, well, it's not arbitrary. There's a reason why they're called 5' to 3', but that's like that's the nitty gritty stuff. That's not super critical just yet, but we'll probably get to that at some point. So yeah, wrapping up some of the primer on DNA, okay, is, well, what happened? How did they go about solving the structure of DNA? Okay, that Watson and Crick. And this is their original publication, okay, in 1953.
Let's go.
And yeah, the molecular structure of nucleic acids, okay. And like on the right, like I pulled this like from the original, like that's literally like what they proposed. They correctly
What's the
proposed.
pole in the middle? Is that just to show orientation?
Yeah, essentially. Exactly. As in that's not actually
It's not an
there.
actual structure.
Yeah.
Okay.
Because again, what we're going to see in about a slide or two, like it's really impressive, at least I think, and I'm pretty sure you'll agree that based off of what they're looking at, for them to extrapolate like this is what DNA looks like, and to pretty much get it like spot on, is like really impressive. But like,
crazy. Oh, it says the vertical line is the fiber axis. I should have just fucking read the thing.
oh no, I don't expect anybody to like, I included it, but you don't need to. But if you just focus on the big stuff, that's pretty much all I want. But yeah, 1953, and like this is actually what they proposed, and again, they were essentially
cool.
they hit the bullseye. And so, getting into like what were they looking at? Well, this is
Oh, the fuck is this?
this is what we call an X-ray diffraction pattern. So they used actual X-rays, okay, and like this is what they they didn't just look at this and draw the figure. There was a bunch of fancy math and equations, blah blah blah. But they use essentially this type of data to like extrapolate, okay, like what the actual structure of DNA was. And if you squint hard enough, okay, if you look in the center, okay, you can maybe, okay, if you squint hard enough, you can maybe like look at, this might be a super outdated reference, like a magic eye. See sort of like not a double helix, but like the sugar phosphate backbone, and like something going in between. Again, you gotta squint really hard, but like this is what they were this is the raw data, some of the raw data they were using to like solve the structure, and it's still in use today, as in this technique of well, at least in the biochemistry world, as I mentioned here in the note, it's still in use today to solve the structure of biomolecules, for example, proteins, okay, and we call this technique X-ray crystallography. Now we're going to be covering in detail what that is, but like it's overall the same process of what was used, 70 years ago to solve the structure of DNA, of using X-rays. There's a little bit of difference when working with proteins, but ultimately it's like, bombarding some stuff with X-rays, and then through fancy, well, now we can do it with computers and all that stuff. But at some, like, they were literally at some point solving equations and doing a bunch of math like by hand. It's actually pretty impressive when you see the math that they're doing. But and introducing it now, I'm going to be emphasizing this, okay, as we move forward. Is like knowing the structure of a molecule, for example, a protein, is critical to understanding its function. There's going to be a theme in biochemistry or just in biology overall, like structure determines function. Okay, that's all. Not only will I be emphasizing the central dogma, I will be emphasizing structure. I'm going to be trying to emphasizing structure determines function. Okay, so yeah, this is what they were looking at. Again, I think pretty impressive.
It's crazy, and I got like I don't know, like a statement slash maybe a question and an
Mhm.
actual question that may not be
Sure.
relevant. Like I just realized that like B. coli, like I've heard of E. coli, like I'm guessing there's an A through at least E, colis. So like I don't know what the fuck that is, or if we'll ever talk about that. And fucking when like who thought or how do they figure out, hey yo, X-rays and let's start shooting these at people and things? Like who,
Uh,
how, like.
yeah, that's a really fantastic question, actually. That now that you brought it up, like once we get to that point, we're going to be covering this technique because it's relevant to proteins. But like, yeah, there's a whole history. Like, well, who would have thought that just putting X-rays or bombarding stuff with X-rays is going to allow us to see what things look like at a molecular level. Okay.
Yeah.
Because there's a history there. It's and well, no surprise, a lot of this is accident. Well, let's just say working with X-rays has the pretty dark history. It unfortunately led to the death of some very impactful and important scientists. But there's a history that like I'm gonna prepare those slides as well. Just as I'll prepare
Sick.
slides on the Urey Miller experiment, I'll
Hell
prepare
yeah.
slides on the history of why X-rays, how was that
Yeah.
discovered. But
Yeah, that's sick.
wrapping up our primer on DNA, and I guess wrapping up this lecture because I want to respect your time. So it ends with a quiz. Okay.
Oh, let's go.
In the structure of DNA, what makes up the backbone? Is it, whoopsies, I can't operate my own slides. Is it cartilage? Is it, adenine and thymine? Is it nucleotides? Or is it deoxyribose and phosphate?
Oh, fucking like okay. So it's the sugar and the phosphate.
You're absolutely right.
And like the nucleotides are the ATCGs, I believe, right? And it's all of it. It's that and the sugars and the phosphates.
Yeah, exactly.
All three of those
Bingo.
things.
All three. Exactly right.
And then then I guess I'm gonna have to say D, but I guess fucking D. DNA is deoxyribose nucleic acid. Okay. So maybe D then? Or is it C? Because, like, C would include the middle parts, which I don't want it to. So D?
Is that your final answer?
I think so. Like, I don't know if if deoxyribosome means sugar, like the sugar, then yes.
You want to
But if
lock
not,
it in?
then
Lock it in for $1 million.
$1 million
Canadian.
bucks. Lock it. $1 million bucks.
All right,
Canadian,
we're good.
tax free.
This constitutes a drum roll.
Oh shit! The pressure.
It's D.
Yes, let's go. Is it called deoxyribosome sugar?
So yeah, it's like
Ribosome sugar?
I wasn't necessarily trying to trick you, but like so I did emphasize sugar phosphate, okay? But sugar's like a broad category of stuff, and I think you made the connection perfectly of like, well, it is called DNA, deoxyribose, and that is it's a little bit hidden, where I think right here, pentose sugar deoxyribose, right there. So you got there, you locked it in.
And it can take multiple forms. Oh my god, that's where the Pokemon Deoxys comes from. Oh my god, dude, that's definitely where it comes from, right?
I'm not super familiar with Pokemon, but it could be.
I need to look into that. Yo, welcome, Wildcat. Great to see you. Oh my god, fuck. Okay, sorry.
No, nothing to apologize for.
Well, like, I was thinking about it, like, when I heard it, like, oh, it's like Deoxys. But then it was, like, the multiple forms and those things. I was like, oh, shit. Okay. Okay, I'm ready for the next one.
No, that's pretty much
it?
That's it. I'm going to, yeah, you absolutely pass it, and just as a brief preview, okay, for next time. So next time, okay, we got our primer on DNA, okay, and next time we're going to get into proteins, okay. So we've covered, when it comes to the central dogma, we've covered, some of the fundamentals of DNA. We obviously sort of got a, I think, hopefully a pretty good idea what DNA looks like at a molecular level. The main components of like your nitrogenous bases. So again, just to sort of, I guess, summarize and wrap up, the DNA is made up of nucleotides. Those are the monomers, okay, to assemble into the polymer of DNA. And nucleotides, as you correctly pointed out when answering the question, like, well, no, nucleotides, they're composed of three things, as I tried to emphasize. It's made of the phosphate, the pentose sugar, the deoxyribose, and the nitrogenous bases, okay? And then, yeah, we're not going to be covering RNA for a little bit. Well, I mean, at the end of the RNA is not super different from DNA. As in, like, let's say, for example, from this figure alone, okay, can you maybe extrapolate some of the difference, at least one difference between DNA and RNA?
Yeah. It fucking lost one of its homies, one of its legs, or its edge, its backbone.
Exactly, as in RNA, generally speaking, is single-stranded, so it's only, one rung of the ladder. As in, if we go back, where, just like, whatever, chop, take the right hand, well, take anyone, but obviously it's more symmetrical on the right, just chop it in half, that's, RNA is single-stranded, so it's just half of that. There's other differences, but that's one of the key ones that's highlighted there in the figure. So, but next time we'll be getting into protein structure and composition, so we'll be talking about proteins, and just as we saw that there's building blocks of DNA, the nucleotides, okay, but the nucleotides, they're made up of the phosphate, okay, the sugar, and the nitrogenous bases. Proteins have building blocks, and they're called amino acids, and we're going to be seeing that, like, next time.
Hell yeah.
And
Fuck
that
yeah.
process, again, just to reiterate, the process, we're not going to see it next time, this translation process, but just to emphasize, translation is RNA into protein.
Sorry, I accidentally hit a button here, and I started playing the fucking Pokedex entry on Deoxys. And Deoxys, like, now that I look it up, is so clearly DNA. Yes, it is a DNA Pokemon. Like, its arms are like fucking DNA fucking double helixes, and it takes multiple forms.
Oh, well, I mean, yeah, that would make sense. And now you know where that deoxy comes from.
Yeah, and honestly, I feel like that shit like's gonna help my weird brain make more sense of these things. Yeah, like, no joke. I don't know why, but it does.
No. I mean, whatever helps. So you're as we learn more and more. Well, you're learning now, but you're gonna see that obviously there's certain advantages of like making connections. Like, oh, this like in my own daily life, in some creative process. Like in Pokemon, someone tried to incorporate, some sciencey stuff that they knew about. And so there's that aspect of like, oh, I can now make that connection. You're going to see that there might be a little bit of a darker side of like, oh, at least for me, it's like I go back and watch, some sci-fi stuff. And, I hear what they're saying. Like, oh, this is like totally like not correct. Or yeah.
Oh, yeah. Well, like Bible Man, one of my favorite shows, like they always do like the most ridiculous nonsense techno babble, and it's just like, okay, you don't even need to be an expert to know that that's nonsense.
Yeah, exactly. And one of
look forward to ruining more.
I'm probably actually gonna try to prepare a slide for this next time, because like this is one of the things that, let's say, ruined the X-Files for me. Okay, where it was like, I don't know, 4 or 5 years ago. I don't know if it's still on Netflix now, but I saw that X-Files was on Netflix, okay? And I pretty much like grew up with that. And so I was like, I'm going to watch it from the beginning. And like I watched the pilot episode, and like within the first 10 minutes, basically Scully is like, speaking her sciencey stuff. I don't know if you've like watched the X-Files, but Scully's like the sciencey one. And she's like, trying to explain like some sciencey concept, and she's like pointing to a figure, okay? And like the figure, like it's clearly pulled from like a textbook, and like it's clear like the figure that she's like pointing, and like the description she's using, like it's absolutely like not like the figure like at all. It's like it
Oh
doesn't
no.
match anything at all as what she's saying. They just pulled like a random figure from like some biochemistry textbook, and then like they just thought that it would pass. At the time, I didn't even pick up on it, but like I just like returned and watched it, and like, oh, I can't watch this anymore.
That's amazing. Amazing.
But
I look
yeah,
forward to learning more, and that's what I want. I want to know when shit is just absolute garbage and nonsense, and not be so easily duped. You know, like not that X Files is necessarily trying to dupe you, but they are. They're trying to make you think that oh wow, this is big science shit. But you know, like people who actually have bad intentions other than just entertaining you and getting you to watch their show.
yeah.
And I think it's very helpful.
Oh yeah,
So
absolutely.
that's
That's also one of my also motivations for doing it. Well, yeah, well, like to hopefully not only just you but other people hopefully have in my own personal experience. Okay, learning about biology and biochemistry, like I feel not fully empowered, but I think I feel quite empowered of like, oh okay, I can navigate at least some things in terms of let's say talking about vaccines, talking about the spike protein. Okay, well we're gonna be talking about proteins, like when we're talking about the spike protein. Okay, well what does that mean? Okay, what is a what is a protein? And then like with that, like you can, armed with that knowledge, like you can clearly, at least for some, let's say anti-vax people that are talking about the spike protein, start listening to them and be armed with the knowledge of proteins. You can clue in pretty quickly that they don't know what they're talking about when it comes to the spike protein.
And that's amazing, and that's great. And honestly, like, knowledge is power, like, meme aside. It genuinely is, and I very much appreciate you. I appreciate the work that you do. I appreciate you spending time to, like, educate me on these things. Make sure to go check out fucking Corey Campbell. I look very forward to doing more and learning more. Because, yeah, it's great. Exactly. Everyone knows mRNA is evil 5G mind control. Absolutely. See?
Absolutely.
And like those are the types of things where like if you know what mRNA is, at least basically you know the very basics that like of like these things. So it's helpful.
Yeah, exactly. And as always, this has been a pleasure, and I always look forward to these lectures, I guess, quote unquote lectures.
Amazing.
And again, I can't thank you enough for agreeing. And again, I'm going to incorporate the feedback of preparing those slides on the Uri Miller experiment, and I think I should have them ready by next week because it's relevant since we're going to be talking about proteins and amino acids. Well, the Uri Miller experiment totally deals with amino acids, so I'm going to try and incorporate that. And then at some point, okay, you asked a really good question of who thought about X-rays and solving the structure of DNA. I'm going to start preparing, or at least outlining those slides when we get to, how do we solve the structure of proteins? And any sort of feedback from you or your audience that you want to provide, by all means, I'll incorporate it.
Hell yeah. My only thing with the X-ray shit is if there's some real hardcore animal shit, just tone her down a bit for me. I can't handle that shit.
Fair enough. Yeah, no, it's same for me.
A casual mention that they did, some animal testing is enough for me. We don't need to go into the details or show pictures of them testing on the animals. I will die.
No, I get it. I'm pretty much on the same page as you. So awesome. with that, I'm going to dismiss the class. Always a pleasure. And see you next time.
Much love. Thank you. Have a great rest of your day, and make sure to go follow Corey Campbell on Twitch. Thank you.
Care.
Bye.
All right, let me switch around 1 second. Okay. All right, well, first, thank you, Orange Hue, for passing by. And yeah, well, so I wanted to, ideally, obviously I couldn't type out a full comment about the conference that, again, I think it's Watson that sort of went off the deep end, or maybe both of them have. But yeah, some of the, I think it was Watson. I think that was I think stripped of their Nobel Prize. I seem to have that in my brain. And thank you, Bob Marley, for passing by. And it's Kieran, and anybody else that I forgot, although I'm not quite done streaming right now. There's still some other stuff that I want to get through. Just give me a moment. Close this. And 1 second. All right, well, that was great, as always. I guess, well, went a little bit longer than I anticipated. I thought not that I'm on any sort of timeline, but I do want to, like, respect, you know, the 1 hour that I, let's say, not that we signed any sort of contract, but, you know, I do want to limit it to 1 hour, so maybe I need to be better in terms of respecting time or honoring, you know, what I agreed to. Although again, this is pretty unstructured, which is actually what I'm going for. I just, well, yeah, I saw the hour mark come up, and I didn't, yeah, I'm learning as I go. I didn't necessarily want to, like, you know, stop in the middle of, like, the DNA stuff. Oh, wow, thank you so much for the raid. Super appreciated. Welcome. Shout out to Talor. Welcome, Wildcat. Great to see you as always. Definitely, you know, if you're not following Talor, you absolutely should be, because, well, we are doing biochemistry lectures, which I'm, again, super excited about. I'm super grateful that he agreed. Yeah, like, that was awesome. Couldn't have asked for anything better, in my mind. And I think it's only going to get better and better from here. Not that I think it's bad now. I'm super pleased with how it's going. But yeah, again, I'm always trying to think of, like, how to make things better, how to improve, okay, the presentation and the slides. And I'm probably going to try to incorporate more questions, because I think it's a good way to, like, reinforce and get people to remember, just as, like, Talor remembered. You remembered perfectly hydrogen bonds. You remembered perfectly covalent bonds. And that, I think that goes back like 2 weeks now. So ace to that. Okay, so where did I want to pick up from here? So, well, my original plan, at least moving forward, is, like, well, yeah, obviously to have the biochemistry lecture stuff, and then to, you know, do my typical research streams, or covering research on stream stuff. But I'm not quite yet at the point of having the time yet to, like, prepare, you know, formal research streams, because, like, yeah, for the past 2 weeks or so, my grandmother has been, like, recovering, a week and a half in the hospital, and so I've been, you know, focused on taking care of my grandmother, who needs, you know, quite a bit of care, understandably, since she's 91. And, well, she deserves also the extra care. But once that stabilizes, okay, as mentioned, I'm going to, again, try to get back into regular streaming. You know, I am regularly streaming now, but it's just, like, once a week on the scheduled biochemistry lecture time. But I hope to increase my stream time to at least twice per week, and have, again, the biochemistry lecture stuff, and then my usual covering scientific research, because there's a lot of scientific research to cover. But there's a little bit of articles that I want to cover today, or now, which is, yeah, I read a misinformation piece last time, and there's this other one that I've been wanting to read. And there's also some interesting research that was published, I think, today, yeah, July 17th, about night owls that, whatever, confirms my bias in terms of night owls appear to have better brain function. I'm personally biased because I'm a night owl and always have been. So, this looks good for me. But I haven't read the research yet. But I'm curious, or I'm even skeptical of like the claim. But we'll see. Thank you so much, Wildcat. At the end of the day, like pretty much, I'm lucky that my grandmother is alive today at 91. Okay, she's gone way past average life expectancy. And so, every day, gotta be grateful for every day, every minute that I have with her. And I'm also, but we as a family are fortunate that despite the fact that she fell, she did not fully break her hip. She didn't hit her head. Okay, she fell while she was in her own residence, semi-assisted residence. But she lives alone in a complex that prepares her meals for her, takes care of the cleaning and all that stuff. But all things considered, despite the fact that she fell, could have been much worse. She needed an operation, but it went well. It wasn't a major operation. It seemed like it lasted a few hours, and even on the day of the operation, like after the operation, after like the sedation and anesthesia wore off, like she was, she seemed to be fully conscious and in good spirits. And she has been discharged from the original hospital that she was brought to, in the emergency room. She's been transferred to essentially a rehabilitation hospital that, there's now intensive physio. But as of like today, she can, take a few steps like with a walker and some assistance. She can sit up in bed. Obviously it's painful and all that stuff. But it's been pretty surprising, at least to me, how quickly she appears to be recovering for someone who is 91 years old. And it's not like, my grandmother, not that she was ever like in bad shape or like out of shape. She always more or less took good care of herself, diet wise, but she wasn't like, a bodybuilder or all that stuff. She wasn't like a professional athlete. But I think that has like worked to her advantage in terms of being able to live to 91. And the fact that, she seems to be bouncing back, I think, pretty quickly, all things considered. So all that to say is like, despite the negative stuff, so far it appears to be in the right direction in terms of recovery. Well, 3:24 like I think, technically speaking, okay, 3:24 a.m., that I don't know if that's night owl territory. Maybe that's more morning owl territory. I don't know how exactly like the chronotypes are like fleshed out. But let's see. So I have the original research here. Sleep duration, chronotype, health and lifestyle factors affect cognition. A UK Biobank cross-sectional study. At 91, my granny passed away a couple years ago at 92. Well, I'm sorry to hear that, but yeah, 92. That's she again far exceeded average life expectancy. We were very relieved she made it through COVID and never had to be in hospital. Yeah, the main thing is being able to be there for them. Yeah, exactly. And that's pretty much essentially is the emphasis now. But an unfortunate reality is like on the topic of like cognition, okay, the cognitive function of my grandmother, okay, like is definitely like on the downward trajectory and has been for quite a few years. Of like, she can converse, she can recall, memories of 50 years ago, but like the recent memory, or like essentially like the short-term or moderate-term memory, like that's essentially gone. She's not going to remember yesterday. She's almost certainly not going to remember like a week ago. And it's, it's basically a gamble whether she'll remember like 15 minutes ago. But like overall, her language faculties appear to be essentially intact, you know, as in like in conversation, she can essentially like converse to a reasonable degree. She can still essentially read. It's slow. But like, yeah, but given the fact that, you know, her cognition is definitely on the downward trend, like she definitely needs some extra supervision. It's as in during the intensive parts of her hospital stay, when she was understandably more disoriented, okay, and didn't recognize that she was in a hospital, that she'd injured herself, you know, she would, you know, try to remove some of the medical equipment. So she needs someone that doesn't need, but rather, instead of restraining her, okay, our family is taking it upon ourselves to ensure that someone's there to make sure at the time that she wasn't, you know, removing her IV, you know, doing things to basically sabotage, well, unintentionally sabotage her care. Obviously, like from her perspective, she's not fully cognizant of like what's going on. She just, you know, she sees an IV and wants to pull it out and all that stuff. So yeah, tricky situation, but at the end of the day, we're lucky still to have her. So this night owl stuff, we'll start here. Gonna get into the misinformation stuff, as in I'm probably gonna read the summary, try to go through a little bit of the research. Doesn't seem super long, probably not gonna read all of it. I'm just mostly curious on how they got their data and how they did their analysis, and then we'll cover this misinformation article that I've been wanting to read for a while. So a person's chronotype defines, let me zoom in, defines what time of the day a person is most active based on their sleeping patterns. For example, are you the type of person who wakes up early and is most active during the morning? Then your chronotype is popularly referred to as a morning lark. If you get the most done at night and then sleep in late, you are considered a night owl. And I think almost certainly I fit in the night owl. I get most of my stuff done at night. You know, I try to be a little bit more reasonable in terms of not going to bed at 3 a.m., although that still happens occasionally. But like, yeah, midnight, 1 a.m. is sort of what I aim for. Past studies have compared these two chronotypes to see how they compare in different areas of health, including physical activity levels and mental, metabolic, and cognitive health. Understanding the impact of sleep on brain function is essential because sleep is crucial for various aspects of our health. National Institute for Health and Care Research, doctoral blah blah blah, explained to, well, yeah. It plays a key role in cognitive function, memory consolidation, emotional balance, and overall mental well-being. Poor sleep quality or insufficient sleep has been linked to numerous health issues, such as increased risk of neurodegenerative diseases, cognitive decline, and daily functioning impairments, she added. West is the lead author of a new study recently published in the journal BMJ Public Health that found that getting between 7 and 9 hours of sleep each night was optimal for brain health. Which I think, you know, that's been the common research finding when it comes to sleep for the past, quite a few years. Like, yeah, aiming for 7 to 9. The research team also found that people who identify as night owls may have better cognitive ability than morning larks. For this study, West and her team analyzed data for more than 26,000 adults ages 53 to 86 listed in the UK Biobank. Participants had completed various cognitive tests, including memory, reaction time, and fluid intelligence. So essentially, I would say assessing generally older or elderly population. The study's first findings that sleeping between 7 and 9 hours each night was optimal for brain function and boosting cognitive ability. Sleeping less than 7 hours and more than 9 hours negatively impacted cognitive test scores. Our finding of the association between sleep duration and cognitive function, with 7 to 9 hours of sleep being optimal, is significant because it aligns with existing evidence and highlights the importance of optimum sleep for maintaining cognitive health. These findings showed that not only too little, but also too much sleep may be detrimental to brain performance. By identifying the best sleep duration, we can improve public health advice and encourage people to prioritize good sleep habits for better cognitive and overall health, she advised. So, next, scientists evaluated cognitive test differences between those who identified as night owls versus morning larks. Previous studies have shown that disruptions in circadian rhythms, like those caused by shift work or jet lag, can negatively impact cognitive abilities, West explained. However, how being a morning or evening person affects cognitive function is less known, especially in older adults, which is why we did this study. When examining chronotypes, the researchers found that night owls, who were more active in the evenings, performed better on cognitive tests than morning larks, who were more active at the beginning of the day. Scientists also found that participants in the middle who did not define themselves as a morning or evening person also had improved cognitive test scores when compared to morning larks. So it seems like the morning people are the worst performing so far. Additionally, West and her team found that younger age participants and those without any chronic diseases generally scored higher in cognitive tests. Not really a surprise. We were intrigued by these findings because previous research on younger people has indicated that morning types tend to have better health and cognitive outcomes. And yeah, that's why, let's say, whatever. There's a part of me that's skeptical on the research because I was aware, like, even though I'm a night owl, some of the research that I've come across is like, well, morning larks or morning owls are better. However, our study suggests that the relationship between chronotype and cognitive performance may change with age. The findings show that evening types were associated with better cognitive function in our older cohort, possibly due to changes in circadian rhythms and their influence on brain function as people age. The next steps could include exploring why evening types might perform better cognitively in older adults, she continued. Investigating whether these findings apply to other age groups and different populations would also be interesting. Additionally, examining the role of sleep quality in greater detail and considering other factors like physical activity, diet, and social engagement would be good, the researcher further noted. Should larks aim to become owls? Some may now worry if they are more morning active than evening active, that they could be harming their cognitive health. Is that really the case, though? According to West, morning active individuals should not worry about harming their cognitive health. Our study found associations, not causality, and there is a wide range of cognitive performance within each chronotype, she emphasized. The key is to focus on getting high-quality sleep and understanding your natural sleep patterns. Maintaining a consistent sleep schedule and ensuring you get the right amount of sleep is crucial for cognitive health, regardless of whether you are a morning or evening person, West advised. Medical News Today also spoke with Clifford Siegel, doctor of osteopathy, not really a fan of, a neurologist at Providence St. John's Health Center in Santa Monica, blah, blah, about the study who agreed with this approach. Get enough sleep would be my answer, and then it doesn't matter, commented Sego, who was not involved in this research. I think definitely people have a tendency to be morning people, or early birds, or larks, or night owls, or be night people. I think it's important to know what your tendency is, but I think it's important to always bookend your activities with enough sleep. In his view, the most important takeaway from the article is to get enough sleep. The study emphasizes 7 to 9 hours of sleep, which he said was what people should focus on. I think it's also important as a neurologist to say if you ever get a sleep debt, you need to pay it off, Segel also advised. So yeah, again, as they say, pretty much an association, but doesn't mean that if you're a morning person that you should become a night owl or an evening lark, whatever terminology that they want to use. But just maybe to briefly go over what they did. I'm very curious. So in the introduction, sleep is a fundamental biological behavior that is universally conserved throughout evolution. However, despite its significance, the function of such a process remains a long debated subject of interest. Emerging studies suggest that sleep plays a crucial role in optimizing cognitive function by contributing to bodily restoration, memory consolidation, learning, and emotional regulation. Sleep impairment, particularly common among elderly people, has been consistently linked to an increased risk of cognitive decline and dementia. Homeostatic immune function is also profoundly influenced by sleep, and sleep impairment has been linked to immune-related neurodegenerative, metabolic, autoimmune, and vascular diseases. Animal studies have further shown that mechanistically, sleep disturbance induces neuroinflammation, complement activation, impaired learning and memory, and affects hippocampus-dependent learning. Population-based research has highlighted the relationship between sleep duration and cognitive abilities, indicating potential cognitive impairments associated with both short and long sleep durations. Severe sleep deprivation has been shown to induce alterations in synaptic plasticity and impairments in learning and memory, thus affecting cognition. Furthermore, the quality of sleep is also a determinant of cognitive performance, where poor sleep quality is correlated with lower cognitive functioning. Sleep provides a restorative and protective function on cognition by the removal of toxic metabolites from the central nervous system. A positive feedback relationship has been suggested between sleep and Alzheimer's disease, whereby poor sleep quality and duration induces amyloid beta peptide accumulation, which in turn also causes poor sleep quality and sleep deprivation, which, in the biochemistry lecture or course that I'm giving, what do we mean by amyloid beta peptide accumulation? Well, we're going to get a better idea of what we mean when we say amyloid beta peptide, because that's related to protein stuff, and we're going to be learning quite a bit about proteins. But beyond sleep duration and quality, the role of circadian preferences or chronotypes and their impact on cognitive abilities are less clear. Despite valuable insights from existing UK biobank studies on sleep and cognition, there is a knowledge gap regarding the influence of chronotypes. Sleep pattern or chronotypes reflects an individual's inclination to sleep at a particular time of the day as a manifestation of one's circadian rhythm. Although studies have shown that disruption in circadian rhythms, such as those from shift work or jet lag, negatively impacted cognitive performance, little is known about how different circadian rhythms or chronotypes, for example morningness or eveningness, are associated with cognitive function. Using the comprehensive data set from the UK Biobank, this study investigates the intricate relationships between sleep duration, quality, and chronotype and their collective impact on cognitive performance. We consider a range of demographic, lifestyle, and comorbidity factors as potential covariates to unravel their influence on the interplay between sleep patterns and cognitive function. Okay, so just a brief overview of what they did. I don't want to get into the weeds of their methodology, but yeah, UK Biobank, so pretty much a database of, a bunch of people and, their health parameters, biomarkers, blah, blah, blah. But it seems like, yeah, that was their data source. They did the cognitive performance was assessed through 4 or 2 cognitive tests designed for the UK Biobank. Okay, so they put through people in their age demographic. I think it was like 56 to 83. Let's see. Cohort 1 consisting of 10,000 participants, mean age 71 years. Yeah, so again, overall older. Cohort 2 comprising a large group of 16,000, mean age 72 years. Completed only 2 of these cognitive tests, but yeah, pretty much basically a bunch of elderly people that were put through some cognitive testing. In the selection of our study, yes, inclusion of a comprehensive sleep parameters. So yeah, they assessed sleep, or assessed, how much sleep these people were reporting. Yeah, complete data across those chosen variables. Okay. Examine the performance of cognitive function. The stability of such ability over time has been established previously. Yes. Okay. Yeah, they assessed fluid intelligence, reasoning, pairs matching, reaction time, memory. So, whatever actual cognitive tests. They assessed sleep variables, sleep duration, sleep pattern, for example, the chronotype, and sleep quality. And sleep duration was collected from the touchscreen question. So it looks like, at least from what I can tell, like they're asking people what they're reporting in terms of their sleep. They don't appear to be going like in a sleep lab and seeing how much they're sleeping, but pretty much asking them how much they think they're sleeping, what their quality of sleep is, and, you know, whatever, categorizing it and all that stuff. Okay, assessed. So the chronotype was assessed through the touch screen question. Do you consider yourself to be, you know, 6 options? Definitely a morning person, more a morning person. So yeah, again, essentially self-report. What do the individuals basically see themselves as? More evening or more morning person? Something in between. They did some, you know, statistical analysis. Not really super concerned with that. Okay, and results, regression analysis, blah blah. Table, demographic characteristics. So yeah, pretty much spans basically 50 to over 80 years old. So again, largely elderly, and other parameters like their other health conditions, blah blah blah. Nothing super surprising. Okay. Yeah, sleep duration. And so what are the overall summary of their findings? Okay. Our study investigated the collective impact of the sleep parameter, sleep duration, chronotype, and quality